In this video, I'm going to talk about cell structures and their functions. First off, cells. We're talking about things that are really small. So how small? Well, if we go down past what we can see with our eye, right, down into this range right here, we're talking about structures that are between 1 and 100 microns, right? So you can see this bacteria is about a half micron wide and about 1.5 microns long. That's a millionth of a meter, right? So there's 1,000 microns in a millimeter and 1,000 millimeters in a meter. So we're talking about structures that are pretty small. Okay, first, two main types of cells, this one and this one. Notice this guy's a lot smaller and a lot more simple, right? He's called a prokaryotic cell. So what is a prokaryotic cell? Prokaryotic cells lack a nucleus, and they also lack other membrane-bound organelles. This big purple fellow over here, he's a eukaryotic cell. So he has a nucleus and other membrane-bound organelles, such as all these things you see over here. Okay. Here's a good example of a prokaryotic cell. Now, prokaryotic cells are not devoid of everything. They still have ribosomes for making proteins. They're still encased by a plasma membrane or a cell membrane. All cells, all living cells have a cell membrane, and all cells have ribosomes. Okay, prokaryotic or bacterial cells also have cell walls. So plants aren't the only cells with cell walls. Bacteria, fungus, and some protists have cell walls as well. And then some bacterial cells have a capsule that helps to keep them kind of safe and protected when they're in a hospitable or harsh environment. It allows them to survive through some adversity. Okay, let's look at an animal cell. So we have an animal cell here. You can see it's got ribosomes. It's got all these membrane-bound structures, making it a eukaryote. But what do you not see in an animal cell that you would see in a plant cell, say? Well, it's not green, so it obviously lacks chloroplasts. Okay, it doesn't have cell walls, so obviously animal cells lack a cell wall. There's little holes in cell walls called plasmodesmata. If you don't have a cell wall, you can't have those holes in it, so it lacks plasmodesmata. And then it also lacks this big thing called a central vacuole. It's like a big swimming pool or water storage container. And a tonoplast, which is actually a component of that. So let's look at a plant cell. Okay, so there's the cell wall. There are those green structures, the chloroplasts. There's that big central vacuole in the tonoplast. And what structures are here that aren't found in animal cells, or that exist in animal cells? Well, we don't see lysosomes. We'll talk about what lysosomes are. We don't see structures called centrioles. They play a key role in cell division. And we don't see flagella in most plant cells. Plant cells don't move a lot. They're immobile mostly, so they're connected by these cell walls. Okay, but we do see a lot of structures that both plants and animal cells, eukaryotic cells, have, like mitochondria, like a nucleus, endoplasmic reticulum. And we're going to go through some of those structures right now. First, one universal structure found in all cells, prokaryotic, eukaryotic, is the plasma membrane. Okay, and the plasma membrane is basically this selectively permeable membrane around cells made up of these little structures called phospholipids. They also have some proteins in them. They might be structural proteins. Some of the proteins are attached to carbohydrates. They're called glycoproteins. They allow for cell recognition. So having type A, B, or O blood depends on the type of glycoproteins that are on your cell membrane. And anyway, so cell membranes, selectively permeable membrane barrier between the cell and its environment, made up of phospholipids, proteins, glycoproteins, and some other structures. We'll get into a little bit more detail when we talk about cell transport and osmosis and diffusion in terms of the plasma membrane or the cell membrane. Plant cell walls. Plant cell walls are made of that important polysaccharide. Do you remember what it's called? Cellulose. Okay, so cellulose is that glucose polymer that makes up plant cell walls. Again, another structure found in plant cells are these little holes in the cell wall called plasmodesmata. One of the characteristics, again, plants have that animals don't are the cell walls and little holes. Plasmodesmata basically let chemical signals, hormones, things exchange between two cells. Obviously, these big walls, you need to have some little gates in there to let things pass through, and that's what the plasmodesmata are. So again, function of plant cell walls is basically structure and support for plants. It's what keeps plants standing upright, are the cell walls. Okay, again, a characteristic of all eukaryotic cells are the nucleus. It's bound by a nuclear membrane, okay, and it contains these threads in here, this material called chromatin, which is also known as DNA. So the nucleus contains DNA, and the DNA obviously controls all cell functions. So the nucleus, we say, is like the headquarters of the cell. Very important structure. Ribosomes are located outside the nucleus. You can see some of them are here on this structure called the endoplasmic reticulum. There they are. They're called bound ribosomes. Some are just free floating in the cytoplasm. They're called free ribosomes. And ribosomes, you may remember from biology, are the sites of protein synthesis. So the DNA makes a copy of itself called mRNA. The mRNA gets moved out to the ribosome, where it gets translated into a protein. So ribosomes are where that protein synthesis occurs. Okay, so those folded membranes located just outside the nucleus are called the endoplasmic reticulum. Endo, inside, plasma, meaning fluid, flexible, reticulum, like a structure or a network of folded membranes. And you can see some of them are called the rough endoplasmic reticulum, some are smooth. The rough are rough because they have those ribosomes on them. The smooth do not have ribosomes, but they have some important functions as well. So the rough ER basically contains ribosomes. Smooth ER contains other important enzymes, such as enzymes that synthesize lipids and also enzymes that destroy toxins. So again, in your liver cells, for example, a lot of toxin-destroying enzymes are found on your smooth endoplasmic reticulum. The Golgi apparatus, so this is usually located just outside the endoplasmic reticulum, and it's basically these series of folded membranes, and this is involved in modifying and packaging proteins. So proteins made in the ribosomes, transported down the endoplasmic reticulum, and then carried to the Golgi apparatus where they might get modified and packaged up. Maybe they'll be secreted out of the cell like insulin, or maybe they'll be modified and packaged into a structure like a lysosome for internal use. So that packaging up of proteins is done in the Golgi body, Golgi apparatus, or we'll just call it the Golgi. Lysosomes, so again, lys means to break up. So we talked about hydrolysis, splitting of water. We've talked about Lysol spray, kills germs, that breaks them up. Lysosomes basically contain digestive enzymes, and they might break up and destroy worn out cell parts. So here's an old mitochondria that's going to be getting fused with a lysosome, and it's going to basically digest it and recycle all the proteins and enzymes and important nutrients in there. Or maybe food within the cell is going to get fused with a lysosome. Or this is a white blood cell. White blood cells have a lot of lysosomes because they're going to be engulfing bacteria and other pathogens, and they need to kill and digest them. So they'll fuse their lysosomes with these engulfed bacteria to digest and kill them. So lysosome is kind of like the recycling center or the stomach of the cell, involved in digesting and recycling of parts. So again, we can see that all these different structures work together, right? So the nucleus contains DNA, which controls all cell activities. The DNA might get copied and send genetic instructions out here to the ribosomes to make proteins. Those proteins might get shipped through the endoplasmic reticulum and packaged up in the Golgi, and then these containers might be sort of secreted out of the cell, or they might remain in the cell, such as a lysosome. So again, this is what's sometimes called the endo or inner membrane system of a cell, and all these different cell structures working together. So here's an example, right? We've got our rough ER. It's going to package up take some proteins, ship them over to the Golgi in a little transport vesicle. The Golgi is going to modify them, turn them into digestive enzymes that will break stuff down, and then package them into little containers. And well, look, we brought in some food out of the cell membrane. If we fuse a lysosome with that food, we can digest it and then have sort of our digested food may be fused out of our cell through exocytosis. Or let's say we've got a dead structure like a mitochondria here. We can fuse our lysosome with that, digest that mitochondria, and then recycle those nutrients. So again, here's some sort of interaction of how these different structures interact together. So the mitochondria, we already talked about that. That's the powerhouse of the cell, right? It's got the inner cristae where the Krebs cycle occurred, the inner membrane, I'm sorry, the cristae are these folds. The matrix is the inside of the mitochondria. The inner membrane where the electron transport chain occurs, and basically what does it do? It converts food energy into the energy in the molecule of ATP. Okay, there's the mitochondria. Plants, again, generate energy from sunlight in the structure called the chloroplasts. So chloroplasts are green because they have chlorophyll in these structures called the grana. Again, these membranes are called thylakoid membranes, and they're involved in photosynthesis. And again, we've already talked a lot about photosynthesis, important process in terms of capturing sun energy. Plants also have this thing called this central vacuole. It's like a big swimming pool, a water storage tank, but it also helps to pressurize the cell. You can think of it also as a little bit like an inner tube in a tire that keeps the cell properly inflated. So these central vacuoles, again, found in plants, serving two functions: storing of water and also maintaining pressure, or what we call turgor pressure within the plant, so the plant doesn't wilt and sag. Then giving the cell's internal structure is the cytoskeleton. Cyto means cell, skeleton, you know what a skeleton is. So the internal structure of the cell provided by these. There are two types of structures that make up cytoskeletons. This thing is called a tube here, is a microtubule, it just means little tube. And then these little threads are called microfilaments. Again, they just means little thread or little filament. And they work together to make up the cytoskeleton. Again, providing internal structure, and then they're going to form this thing called a spindle. Remember, during mitosis, the spindle forms, and we have chromosomes line up in the middle, and they move to each end. That's an arrangement of the cytoskeleton that provides that spindle structure for cell division. Okay, other structures of microfilaments. Well, in muscle cells, these filaments pull against each other and provide for muscle contraction. So there's another function of microfilaments. They can also be involved with cytoplasmic streaming. Basically, they can create a current in here and allow for movement of nutrients or mixing of things within cells. And they can also work together to kind of push against the cell membrane and allow something like an amoeba to move through the environment. The cytoskeleton kind of providing an inner skeleton for the movement of the amoeba. So some examples of work of the cytoskeletons. Microtubules also make up these important structures found in animal cells called centrioles. Again, the centrioles are going to form an anchor in the spindle for cell division, found in, again, animal cells, not plant cells, but they're, again, important in cell division and anchor the spindle during mitosis. Okay, and then we have some microtubules or microfilaments that are found outside the cell. So this top one, that's obviously a sperm, but remember the name of the little tail of the sperm? It's called a flagella. I don't know if you can see that. My head's probably in the way. The smaller, shorter little things that all beat together, these little filaments here that beat together, are called cilia. So one long tail is a flagella, many short ones are cilia. Okay, and that is it. We've reviewed all the cell structures, and we'll work and kind of talk about these in class a little bit later.